Thank you for inviting me, and thank you to Dr. Donnelly for a great introduction. So I'm talking about a more project-oriented application for the Axiom Array. I'm from the Geisel School of Medicine, which you may know Geisel as Dr. Seuss, so sort of the cat and hat School of Medicine. But anyway, background is that, in terms of this project, we know that genetic factors contribute to lung cancer risk, somewhat unpopular among the sort of environmental epi people, but there's a interaction between genetic and environmental factors that are quite important. If you look among condition on smoking, the odds ratios are about 2 to 2.5 among smoking individuals for increased risk for lung cancer based on family history. And there are certain high-risk factors, like if you have a retinoblastoma mutation or a P53 mutation, that greatly increases your risk. The other side of this is that there are many well-known candidate genes that are thought to be relevant, like cytochrome P451A1 or cytochrome P452A6. The metabolic types of genes often have indels or repetitive elements near the promoters that make them quite challenging to genotype on many SNP arrays. The Axiom array has more flexibility, so that's a benefit. There have been several genome-wide scans that have identified genetic factors. I'll talk briefly about those. So our studies organized in the way it's shown here. The discovery component, which I'm talking about now, has sort of 3 waves. The initial one is integrating genome-wide scans. The second wave was to perform fine mapping for the variants that we'd identified. And initially, that was all we were really meant to do. And then the last wave is replication. And in the fine mapping component and the replication, we were focusing on cohort studies. The cohorts often have samples that have been collected sometimes decades into the past. And some of these I'll talk about later were on Guthrie cards and things like that. So I can share with you some of the results that we had from these fairly heterogeneous collections. And just to tell you that lung cancer, while clinically may be viewed as one thing to some extent, at least actually it's quite different in terms of the genetic makeup, and the predisposition is also very different. So adenocarcinoma typically is less influenced by smoking behavior. So going into this study, we might have thought that there would be more genetic signals, but we don't see that. Perhaps it's more heterogeneous in its genetic background. On the other hand, squamous carcinoma, we see quite a large number of different signals, including in the HLA region, which Peter mentioned is challenging to genotype across. And for smoking behavior, the most significant region for lung cancer and smoking is this region on chromosome 15, which includes nicotinic receptors. As we've done these genome-wide scans, that was the most significant region. It's a very relevant area to focus on. And we have done some sequencing in the region. This is an earlier study we did, which identified where we just sequenced the exons. What you find when you do that is that there's quite a tremendous amount of variability in this region, not just the common variants, but multiple rare ones. And in addition, there's a 22 base pair insertion deletion in the promoter of nicotinic receptor alpha 5 that influences the expression levels. There's an estrogen receptor binding site, and so if you look in females, there's a stronger risk from this allele. It's again very difficult to genotype on a standard platform. So we had been using gels, and that's not very easy to do. So at the point where we were meant to do a sort of a replication study of, and this was meant to be using a bead express platform So that when we wrote this grant, that was all we could afford. And by the time we got to Affymetrix, we could suddenly afford to do 400,000 markers for the price of 384 or whatever. So that just shows you the great benefits of the technology as it goes forward in time. We couldn't do many more people than we planned, so we have about 7,500 cases and controls. And one of the newer observations we have is that at least 2 of the SNPs that we've been able to identify from our studies are rare variants. We have a paper submitted to Nature Genetics on this topic. So these 2 rare variants have about one of them has about a 2.5-fold increased risk. The other one has about a 0.6, I think it is, decreased risk. So these are, items that you really would like to be able to genotype in your analyses. They're not covered well by the GWAS platforms because of their rarity. So, we were excited when we had the opportunity to use the Axiom Array to genotype our samples. What we chose to pursue is a platform with the standard 300,000 exome markers that are included, as well as 100,000 custom content. So that was less money than we had in our original proposal for 384. Of course, they keep chipping away at your money every year. So, even though it was less, we had enough to do what we needed to do. Then the Exome Array had rare variants, which we find to be quite useful for our disease. And the flexibility in the design allowed us to include the small indels. In particular, the 22 base pair insertion deletion in the promoter of alpha-5 was very important to us, and we were able to cover that. We also were able to cover CYP2A6 much more completely than we had been in the past, again, because of the flexibility. And our selection criteria for the 100,000 markers, included a pretty broad range of inputs. So we had the meta-analyses that had been done, so we accepted all the SNPs from that were less than 10 to the minus 4. We also had GWAS in other populations, some pathway-based analyses. Then we did sort of tagging and fine mapping for certain regions. We actually got quite a lot of help from the Affymetrix consultants in designing our platform for these regions. And then we had candidate genes, rare variants from some sequencing projects, EQTLs. We also got input from people that study COPD and asthma, inflammation. So it's a pretty broad reach that we were able to do. In this phase of our experiment, we're receiving samples from 5 different centers. They're all shipped to University of Toronto, and we had planned to have spot assessment, to have the first plate from each site completely checked for DNA concentration and for ladders to be done, and then after that to just do spot checks, 1 row per plate. And the Australian collaborator had samples that had been stored on Guthrie cards. Some of these were up to like 15 years old. So we also did a pilot study on that. So this is the way that most of our data looks. Most of the time it's supposed to be sent 10 nanograms per microliter, and there's a little bit of variability from our good groups that have good quality control. And the gels look very good. Then we have some other groups where the cohorts are older, where there's a little bit more of a challenge. And in particular, this group kept sending us samples that were over-concentrated. So instead of 10 nanograms per ml, we were getting like 10 nanograms per microliter. We were getting 5 times as much. So we tried just cutting the concentration by a certain amount overall, and so that worked for most of the cases, but sometimes it was too low, and so we had a few failures there. The gels look good. And then on the Guthrie cards, we had more failures, but actually the failure rate was not actually too bad. If you look at the gels, they look horrible. Actually, this performed much better than I would have imagined, given the ancient nature of the DNA. Having said that, though, we decided to not do all the Guthrie cards and save the DNA for spot genotyping, mostly just because the failure rates were a little bit higher, and we decided to save the resource for better quality DNA. So in terms of where we are in this project, we're about halfway through at this point. Among the initial markers that we did, among the samples that we've done, we have 53 samples out of 5,000 that were included, including some of the Guthrie card samples that didn't behave very well. So actually the failure rate's quite low. And for the markers, again, we have about 98.5% past the call rate filter. So again, in considering that there's quite a lot of custom content on our platform, this is a pretty good QC. In terms of minor allele frequency, remember this is an exome array, so there's quite a lot of markers that have very, very low allele frequencies, or it may end up being not polymorphic. So that's sort of a challenge with exome arrays, is that you get quite a lot of territory that's not that informative. The missing call rate has this sort of strange bimodal distribution, but you can see that overall, it's very low. And the other thing to mention is that, we're down here doing a GWAS, and we're doing some exome content to try and get a little bit of a handle onto this sort of low effect and somewhat uncommon, moderate effect, but uncommon variance. So that's usually the area of sequencing. And one of the great benefits of having included the exome array is that we were able to compete for another grant in which we will now sequence 1,000 lung cancer cases and 1,000 controls for the exome and the targeted regions that were of interest. So our initial emphasis was not on the exome content, but having it available ended up being sort of a big plus for us in being able to cover a lot more territory and do a much greater review of the genetic susceptibility to lung cancer. So in conclusion, our large-scale GWASs have identified many candidate loci. The known regions contain genotypic features that we know can be difficult to assay, and so we're very happy that we're able to use a platform that's a little bit more flexible. We still can't cover some of the regions as completely as you might like. For instance, the alpha-3 region contains a sort of a microsatellite, and it's impossible to genotype that on any most high throughput platforms. So that's just an overall limitation. But aside from that, the Axiom array was very very useful for us, and there has been flexibility, including these other SNPs and the low cost. And finally, the exome, for us, the exome content was great. It was a very useful thing to have.